Okay. Thank you, everybody, for coming. Thank you to Mary, to Liz, and the QAAFI Seminar Series group for inviting me to give a talk today. As Mary indicated, I hail from Saskatchewan, Saskatchewan, Canada. I realized that maybe I should have put a few slides to kind of talk a little bit about the institute, the university I'm from. But Saskatchewan is basically the third province from the west in Canada, very much known for agriculture. Saskatoon's about 6 hours from Calgary. Most people in Australia know where Calgary is. So 6 hours basically east of Calgary. And I'm part of a large agricultural college at the University of Saskatchewan, specifically the Animal and Poultry Science Department. And while my background is toxicology and animal physiology, that is what my early training was in and my graduate work. Joining the agricultural college at the University of Saskatchewan allowed me to meld my animal physiology and toxicology, mechanistic toxicology interests with a current regional as well as global issue in animal agriculture, as well as crop agriculture, which are mycotoxins. So that's what I'm going to be talking to you about today. So I like to actually start with this slide, a citation from the Food and Agriculture Organization of the United Nations, basically indicating that there's an ongoing need to protect the health of humans and susceptible animals by limiting exposure to mycotoxins. And even though there's a lot of research and development focused on this area, mycotoxins continue to be a global issue. And so for my talk today, I'm first going to briefly introduce mycotoxins in both the global context as well as with a particular focus on the situation in North America, where kind of the research that I'm doing has relevance in terms of the type of mycotoxins I'm looking at. And then talking about moving on to impacts on agriculture with specific research out of my group on poultry. I also do mycotoxin research with swine. So I mostly focus on monogastrics, although I know there's a lot of really interesting, specifically microbiome research with ruminants these days. And then I'm just going to talk a little bit about mitigation strategies with a really exciting study that just came out that I was part of with looking at alternative uses for what we call garbage or salvage grain that has no value, highly damaged by fungus, and has high levels of mycotoxin. So basically looking at, can we use this garbage grain, this no-value grain, and feed it to animals, insects specifically, to potentially produce a high-quality protein source that could then be utilized in animal feed. So first, what are mycotoxins? Just as a brief introduction, although I'm sure many in the audience are very familiar with mycotoxins. They're basically the secondary metabolites of fungi that colonize plants, plant products as mold, and can either occur both pre-harvest or post-harvest or into storage conditions. Mycotoxin contamination is particularly complicated because one fungal growth, one fungal species, can produce multiple mycotoxins. At the same time, grain can have different type of fungi on the grain, and they can produce the same or different types of mycotoxins. So mixture toxicity, when we're talking about a toxicity context, is the rule, not the exception. We have multiple mycotoxins in one sample. And the other thing about mycotoxins is that they're not usually eliminated through feed and food processing. They have great stability against heat, physical and chemical treatments. So they're not often eliminated, most likely not eliminated in feed and food processing. Right now, according to current literature, there's about 300 to 400 different types of mycotoxins identified, with more becoming evident as new isolation and analytical techniques become refined. And even with a high number of different mycotoxins identified, there are a few common ones that regularly contaminate food and animal feed products. One which most people are familiar with is aflatoxin, probably the most studied and most toxic mycotoxin, responsible for carcinogenic, teratogenic, hepatotoxic, so liver toxicity. Liver is a primary target. Mutagenic, also immunosuppressive effects, so suppressing the immune system. So we see aflatoxin up here. Zearalenone here is really structurally similar to naturally occurring endogenous estrogen. So it's often attributed to reproductive issues, infertility in animals, because it's able to mimic the actions of estrogen. Deoxynivalenol, or DON, is the one here. And it's probably, well, it is the most predominant mycotoxin worldwide, no matter what continent you're referring to. The most common symptom of DON in animals at low levels is poor performance, poor growth and performance, which makes it really hard to determine if that you do have DON contamination in animal feed, because there's a lot of things that you can attribute in animal production to poor growth and performance. Other factors associated with DON toxicity is lowered milk and egg production, as well as intestinal damage, as well as immune suppression. So it actually can be attributed to immune impacts, immunotoxicity, reduced vaccine response, etc. So this mycotoxin risk map is from 2017, and these risk maps are often released by Biomin. And it basically looks at mycotoxins compiled for each region, mycotoxin occurrence, and basically risk levels of different mycotoxins according to different regions of the world. And so the risk level is expressed as percentage of feed samples that are testing positive for at least one mycotoxin above what's considered the threshold for risk. And so the red color here in Canada, China, for example, this is specifically for 2017, indicates that about 3/4 to 100%, so 76 to 100% of samples were above the risk threshold for a particular mycotoxin. So that's quite high, obviously. North America here specifically faces, if you look, it's really hard to see the specific mycotoxins, but DON, fumonisin, are specific mycotoxins that were the ones that were primarily above this risk threshold. And more than 80% of tested samples overall tested above the risk threshold in North America. The most frequently detected, like I said, is DON. And the most single highest DON concentration worldwide was actually detected in North America, and that was at about 51,000 ppb, or 51 ppm. So that's extremely high. The risk threshold, according to Biomin for DON, is 150 ppb. So obviously 100 times greater than the risk threshold. So to kind of bring back to our status or the context of North America and a particular mycotoxin, Fusarium, fungal fungus that produces mycotoxins in North America, Fusarium, they're an extreme problem in terms of mycotoxins worldwide, and several Fusarium species are responsible for a disease in different grain products called Fusarium head blight, specifically in wheat. So this is a disease caused by infection with Fusarium. What you see typically is these kernels that are shriveled, chalky, and pinkish. These are called Fusarium damaged kernels. They're also called tombstone kernels. And this is due to the damage that the Fusarium fungus does to the wheat itself. And within the wheat, it's producing these secondary metabolites, specifically deoxynivalenol, and I'll come back to that in a minute. Fusarium graminearum, specific species of Fusarium, are most common in Western Canadian wheat. And this is actually a sorted wheat sample. These are the healthy, kind of plump, darker kernels. These are the more shriveled, kind of lighter, chalky kernels. So you can see physically the difference. This is a sorted sample, so it's been concentrated. So it's easy to see the distinct difference between these two samples, Fusarium damaged and healthy kernels. If this is a sample with a lower percent of Fusarium damaged wheat, that difference is not quite as apparent. So Fusarium damaged kernels, FDK grain, is associated with a lot of characteristics that are not sought after in terms of crop production or for feed or food as a feed or food commodity. Lower yield, lower quality as food or malt, and with that comes lower grade and price. So if we look at percent Fusarium damage, this is Canadian Western Red Spring wheat. 0.25% damage within a sample, so of Fusarium damage within a sample that would be identified by graders is grade 1. So this would be the highest value wheat. As you increase in percent Fusarium damage, you get to 4% Fusarium damage, and you're at feed grade wheat. So lower value, but still accessible, still acceptable for manufacturing of animal feed. When you get to salvage greater than 10% Fusarium damage, this is no value. This has zero value for the producer, and there is really nothing that they can do unless they want to blend that with and purchase the higher grade wheat, or they bury it or burn it. So that is basically the option for that type of wheat. The difference between number 1 and feed grade in terms of price is typically about $100 per metric ton. So about 30% difference in price. So there's a significant difference, while obviously salvage has no value. And overall, there's been about $5 billion in losses due to Fusarium damage or mycotoxin grain realized annually in Canada. So this is a huge loss to, obviously, crop producers. And just to kind of put Canada in context in terms of the world wheat production, about 727 million tons of wheat are produced annually, and about 15 to 20% of that in a worldwide context is wasted per year. In Canada, 55 million tons of wheat are produced annually, and in Saskatchewan, the province that I'm from, 55% of that is produced in our province. So we are very the most prominent grain producers within our country. And 10 to 15% within Canadian wheat is considered unsalvageable, so garbage. It has absolutely no value. So obviously the quality of the wheat goes down with Fusarium damage, but the other thing that in terms of animal health, in that feed grade wheat that still has Fusarium damaged kernels is the relationship between Fusarium damage and mycotoxin concentration or levels. So this is actually a paper by Cheryl Tittlemeyer, a colleague of mine. She's with Agriculture and Agri-Food Canada in Manitoba. And this was published very recently, basically showing the relationship between Fusarium damage and DON in Canadian wheat. And so this is actually various wheat varieties from the 2016 growing season in Canada, which was a very bad growing season, one of the worst Fusarium years in recent history. And you can clearly see that there's a significant relationship with the percent of Fusarium damage in a wheat sample and increasing Deoxynivalenol concentration, the specific mycotoxin. And so this is quite clear in terms of the relationship. And so it's fairly easy, it's fairly safe to say that if you increase the Fusarium damage within wheat, you will have higher DON concentrations in that sample. And I'm going to be talking a lot about DON. DON is basically one of these mycotoxins within animal health that can have various negative or adverse effects. Effects on growth performance, like I said, immune and gut health, as well as reproduction and hormone physiology. endocrinology. We see losses in performance, increased health costs, and potential condemnation of feed or animal products. And a significant portion of animal production costs is due to feed costs. Farmers are often tempted to purchase this lower value grain used in animal diets in order to reduce feed costs. And as the increase in Fusarium damage and increase in mycotoxin in particular growing years, and it is increasing on average due to various factors, there'll be more of the lower grade, lower quality, higher mycotoxin feed being shuttled over or shunted over to animal production, while the higher quality grain will be saved for human consumption. This practice of increasing the amount of mycotoxins in animal diets, unfortunately, carries risk of not only from mycotoxin contamination, but in some cases it can also alter nutrient composition. It means lower protein often in feed. So all of that can contribute to poor performance in animal production. So I'm going to talk a little bit about some of the research that we're doing related to mycotoxins in poultry health specifically, to kind of better understand factors that are associated with animal susceptibility, sensitivity to mycotoxins. And the question is, why poultry? Poultry, if you actually look at ranking of animal production species, poultry actually rank as among the more insensitive to mycotoxins and specifically DON. And swine being more sensitive, swine is among the most sensitive to DON in animal feed. Right now, 5 milligrams per kilogram or 5 ppm is currently the dietary allowance for poultry through various legislations. And 1 milligram per kilogram is allowable for swine, even lower for younger animals. And so why are we concerned about poultry? Well, modern broiler chickens are selected for superior performance, but require high quality diets to reach that genetic potential. And this has changed over time. And so there's actually a lot of inconsistency in results obtained across the last several decades in poultry studies, because the allowance level, the guidance levels for poultry of 5 milligrams per kilogram threshold is actually in some cases attributed to adverse outcome in poultry, poor performance, poor immune response, poor vaccine response, and in other cases it's not. So it's kind of in that threshold level that it may need to be revisited in order to be appropriate for modern broilers. The other relevance for industry is the fact that poultry industry, as many animal production industries, are moving towards antibiotic-free poultry production, which requires the ability to achieve the same growth performance in a cost-efficient manner under an antibiotic-free system. And so it's really important for us to understand how different factors can influence the effects of mycotoxin, including DON, on poultry performance and health. And this will also hopefully help us direct potential methods to mitigate these adverse effects. So one of the students in my group, Anhao Wang, he finished his PhD, defended his PhD in December, and his PhD thesis was focusing on several different research objectives, among them to identify factors that influence susceptibility of poultry, and he focused on broilers, to mycotoxins, develop sensitive methods for evaluating exposure and toxicity and adverse effects in poultry. One of the things he focused on was behavior, which is really neat. And also he tested a few different strategies for reducing exposure and negative impacts, toxicity of mycotoxins in poultry. So one of the studies he did, let's check my time here. One of the studies he did was look at how timing and exposure duration during a typical broiler production cycle affect the outcomes of mycotoxins in the diet. So specifically naturally contaminated diets. So these are diets that are not spiked with one mycotoxin, but they're made with Fusarium damaged grain, which provides a more industry relevant exposure scenario. And he wanted to look at if there's a particular period in the broiler production cycle where mycotoxins were the most detrimental to production and other endpoints that he looked at. So he used a Fusarium damaged wheat, 2 wheat sources actually, that he got from grain producers in Saskatchewan. One was very highly contaminated, high levels of Fusarium damaged kernels, and one was a relatively clean sample. He formulated diets at the Canadian Feed Research Centre, which is about an hour north of Saskatoon in North Battleford, Saskatchewan. And this is a full-scale, both industrial plant as well as it has a pilot and research line. So we can actually make diets with the same feed ingredients and the same processing technologies at a smaller scale to do studies such as this, which is great. It also allows the grad students that are involved in these studies to actually go up and be part of the diet formulation process. The dietary mycotoxins were not only analyzed in grain prior to making these diets, but they also were analyzed in the diets prior to feeding, just to confirm. Our mycotoxin analyses is done across the road at our vet college as part of a private diagnostic testing lab, Prairie Diagnostic Services, and they use HPLC MSMS for that technology, and they look at about 14 to 16 different mycotoxins, either in grain or in feed. So the diets he ended up getting from his formulations, which he aimed for, was a control diet, low levels of DON, well below kind of threshold levels for poultry, but obviously still had some levels. It's basically impossible to get a fully zero mycotoxin diet. And a DON-contaminated diet that ranged from about 6.5 to 7.5 milligrams per kilogram. So right around that 5 milligram per kilogram threshold for poultry. His feeding program, I'll just kind of take you through it. This is a schematic representation of his feeding program starting from hatch to 34 days. Consisted of both a starter diet and a grower diet, which is why there was that range on the previous slide of DON contamination in the diets. And so basically he had one treatment where it was fed the control diet throughout the entire production series. And then basically taking part of the production cycle here and giving DON diet, the mycotoxin contaminated diet, at specific parts of the production cycle. And he wanted to see, by measuring different performance parameters as well as other parameters throughout this period, where kind of the adverse effects start showing up in these different types of, in these different time series. And of course he had a DON contaminated treatment that went right from day 1 to day 34. So this is some of the average daily feed intake data that he obtained for the 1 to 14 day period, the 15 to 21 day period, and that last grower phase, which is the 22 to 34 day period. And average daily feed intake indicated here on the y-axis. And so we really didn't see any effect of that early grower diet, early starter diet at the 1 to 14 day period. Here we start seeing a bit of a drop in performance, and again, this darker bar here is DON 1 to 34 days. So these would be animals that are fed the DON contaminated diet for the full 21 days. And we're starting to see a drop in average daily feed intake here. And then if we look at the average daily feed intake here of animals that are fed the DON contaminated diets either just during that grower phase, or I should say here, the second from last bar, or from ones that are fed the diet throughout the entire period, this drop in feed intake is equal. So the birds had equal drop in feed intake whether or not they were fed throughout the entire production period or whether they were fed just during that latter production period. So obviously this drop in feed intake is more pronounced during the grower phase than it is during the earlier growth phases. And this was actually mirrored in the average daily gain as well. During the first 2 weeks of the trial, average daily gain was not affected by the presence of mycotoxins or DON in the diet. However, when assessed at day 21 here, we saw average daily gain was decreased for birds consuming DON-contaminated diets compared to the same age birds taking in the control diets. And from 22 to 34 days here, birds consuming the DON-contaminated diets both through the growth period, throughout the entire growth period, and only during the grower phase had equal decreases in average daily gain. And when he looked at the gastrointestinal tract at both 21 days and 34 days, he actually saw a decrease in villi height. So here we have villi height and crypt depth, kind of below the 0 x-axis and on the y-axis, and villi height above. So if we think about villi being these long finger-like projections in the intestine responsible for site of nutrient digestion and absorption, and obviously necessary for a fully functioning gut and a fast-growing bird, we see that with the DON contaminated diets fed throughout the entire production period here from 1 to 34 days, we saw a significant decrease in villi height. These are the DON-contaminated birds here. And we also see a shallowing of the crypt depth. So obviously, there is associated effects on the gut when we see effects on performance. This has been associated in other studies with reduction in nutrient digestion and absorption. We didn't measure specifically nutrient digestibility in this study, but I would say the effects on the intestine, as well as reduction in feed efficiency, feed intake, and average daily gain, is enough to suggest that there's impaired nutrient digestion and absorption in these birds. So what he found basically from this first study of his thesis, and I'm not going to go through all of his studies, he did many, was that broiler chickens are more sensitive to dietary Fusarium mycotoxins during the later stages of growth, and that this reduction may be the result of altered intestinal mucosa and absorptive structures, specifically in the ileal region of the gut. But we also see a decrease in feed intake. So there may be an effect of feed refusal, decrease in feed intake with the presence of these mycotoxins. And this allows us in future studies that he did, to basically refine the experimental period. So we actually then followed up to answer a question of what is the predominant mechanism underlying decrease in feed intake associated with Fusarium mycotoxins or DON in the diet, and growth suppression. So one of the things he did to do this was look at behavior. So he wanted to answer the question, can decreased feed intake be attributed to changes in feed preference and changes in feeding behaviors in birds that are fed DON-contaminated diets? And because we were able to refine the exposure period to the grower phase, to that later stage, he was able to just focus on behavior, not during 34 days, but just during that period, which was much more logically feasible. There's a lot of video analysis being done for this study and allowed him to focus on older birds. So we did 2 feeding trials, feed preference trial and a feeding behavior trial. And what he did, he actually fed a corn-based diet for the first 21 days. So this allowed the animals not to get habituated to a specific wheat-based diet. And then he provided them from 21 to 27 days, kind of a 1-week period. He gave them diets that were either control diets with low DON-contaminated material, low DON diets, so this is actually half of the threshold for DON and poultry, and then a high DON diet, which is around that 5 ppm, 5 milligram per kilogram threshold. And again, DON was the mycotoxin that was the predominant one in these studies, so that's why I'm referring to these as DON diets. I should have said that earlier. The feed preference trial, what he did for this was use Ross 308 birds, and he had birds set up in 5 cages per treatment, 4 birds per cage, and the feeder that was in front of our battery cages was split. Birds in that cage had access to 2 different diets that were placed in that feeder. The combinations were control and low DON and high DON, or control and high DON. And so basically what he did was he recorded daily and overall feed consumption of each diet per cage. And so this didn't require any video analyses. There was no video on this, but the behavior study that followed this had the video analyses. So when he looked at the data, he looked at the proportional feed intake each day throughout the from 21 to 27 days, the 6 or 7 day period. And what he saw was that when given a choice, broilers exhibited immediate, so this was interesting, right away, when given a choice between the control diet and a low mycotoxin, or the control diet and a high mycotoxin DON contaminated diet, they showed a clear preference to the control diets over the mycotoxin contaminated diets. And this isn't something that they kind of learned the more they fed. It was something that as soon as that diet was introduced on day 21, they immediately consumed more of the control diet with an aversion to the mycotoxin at both levels of mycotoxins. And this, if you remember, this is at levels, the low is at levels that are below the threshold set for poultry. So immediate and prolonged aversion to DON contaminated diets. And there was no difference. There was no difference when given the low and high, when given the choice there. So there was no distinction between the mycotoxin diets at the 2 different levels provided. When you looked at overall feed preference, when provided a choice, overall, broiler chickens preferred uncontaminated diets over DON-contaminated diets when given free choice. It's pretty clear that when given a choice between control and low, on average, they consume more of the control diet as compared to the mycotoxin or DON-contaminated diet, again, with no difference in overall average daily feed intake over the 6 to 7 days. So for his behavior trial, what he wanted to do was look at whether the behaviors, the feeding behaviors of the animals differed with the different diets. So again, this was Ross 308 birds. In this case, every cage got a particular diet, so the feeders were not split into two different mycotoxin diets, DON-contaminated diets, and the behaviors were recorded, both at feeder behaviors, so feeding, and at drinker behaviors. And birds were marked on their head with markers so that they could be identified through videotaping through the front of the cages. So this was a lot of sitting in front of videos, post-experiment for ANHOW, and looking at recording behavioral observations. And this was verified by a second student in our animal welfare group who does broiler behavior analyses, just to make sure that the findings, what he was measuring, was accurate. And so what he did is he looked at, if you look at the feeding behavioral design, basically there's, if you look at a typical day here, period, 24-hour period, we have a light period here with a dark period in the middle. And so what he did, this would be the beginning, the morning of feeding. And so what he did is he did 1-hour video recordings, the first one right before the dark period set in. So there was an hour of video recording here before the dark period, an hour of video recording at the end, at the beginning of the light period, and then one right before feeding. And so he looked at all these 3 1-hour periods to measure feeding behaviors. And what he found overall, that regardless of the video recording period, whether it was early in the that light cycle, halfway through, or right before the dark period, regardless of the observation period, the animals that were given either the low or high DON diets had spent more time at the feeder compared to the control diets. So there was a longer, what we call at-feeder bouts. They spent longer at the feeder than the animals fed the control diet. So this could be due to various underlying mechanisms, but basically it took longer for the animals to eat the feed at every visit to the feeder when there was contaminated diet provided compared to the controls. And with this, we also saw during, again, to back up what he saw during his developmental sensitivity study, he saw effects at feed refusal and growth suppression, but only at the high animals. Only the high mycotoxin animals caused that growth suppression. He didn't see it at the low DON diets, although he did see behavioral effects. So he saw that the effects on behavior actually are able, you can actually discern DON at lower levels with behavioral effects than you can with performance effects. So that's what this indicates to us. So his work has kind of started the conversation, at least with poultry producers in Canada, that it may be prudent to revisit guidelines for poultry, DON in poultry feed. And there's reason to believe that we could see performance effects as well as adverse effects at lower levels than we previously would have expected. Okay, so I'm going to switch gears a little bit to this new study that came out that was just published in the journal Toxins about a week ago, looking at mitigation strategies for mycotoxins. And just as a quick highlight about mitigation strategies, pre-harvest control strategies are associated with things like changing crop management practices, work on plant genetics to improve resistance to fungal colonization, and biological control methods. Post-harvest control, which is what most of the work that we're involved with, some of it involves sorting technology, so looking at different ways to sort grain effectively and at high amounts to scale up to industry relevant practices. The addition of detoxifications or binders along this line, not necessarily something that binds up the mycotoxin, but something that may improve the gut health of the animal, so it may become more resistant to the toxicity of mycotoxins is another way. To kind of post-harvest control or reduce the toxicity of mycotoxins. But another way that we're kind of looking at is looking for alternative uses for this salvage or waste grain that is being produced as associated with Fusarium damage and high mycotoxin content. So this is some kind of a unique way that we may be able to utilize this grain, provide a market for this grain, in this case wheat, in order to actually produce something that is viable and can be used in the animal feeds industry. Because right now this grain cannot. So what if we feed it to animals that actually can tolerate it, that can detoxify it, that can grow, and not carry a body burden of these mycotoxins? And what if these animals are insects and may be able to be used downstream as a viable alternative protein source for animal feed? So insects have been, part of the diet of humans and animals for a very long time. Insects have the capacity, and there's a lot of focus now on insects that have the capacity and using them to recycle low-value organic material, including waste material, bio-waste material, and convert these animals, these insects, into a source of protein. This won't work if the animals actually carry a body burden of these mycotoxins, if they then become a contaminated commodity. Specifically, there's been studies with yellow mealworm larvae, and they actually, in some of the recent studies, it's been shown that yellow mealworm larvae actually prefer wheat kernels artificially infected with Fusarium species. And then these yellow mealworm larvae, yellow mealworms, actually have a fairly high crude protein and crude fat content that makes them very amenable to be utilized in feed production. So one of the research objectives of Carlos, a master's student on this project, was to determine whether yellow mealworm larvae could be reared on mycotoxin-contaminated wheat to produce a safe and efficient alternative protein, source of protein for animal feed. And what Carlos did was he, again, took different sources of wheat, confirmed that the fungal growth on these wheat was Fusarium germinarum, and then took these sources of wheat, sorted them using a bow mill NIT or NIR sorting technology, and produced two different fractions, a high and low quality fraction, and then recombined them to get different levels of wheat for diets for these worms. This doesn't have to be formulated into feed after this. This can be then crushed and provided to the mealworms as their food source. What he did was he did a feeding and breeding trial. Afterwards, he took the mealworms, weighed the mealworms, and then also had them analyzed. And this took some optimization for mycotoxin and nutritional analysis. And just to highlight a few of his main findings, after 4 weeks on the DON contaminated diet, so low control, low, medium, and high, what was found was that the mealworms analyzed for the same mycotoxins actually had very little and equal to the amount of DON present in the actual tissue of the mealworms after 4 weeks of feeding them the DON contaminated diets. We could find a derivative of DON, 3A DON as well, again at very low levels. And what we found, frass is actually the waste material from mealworms. I didn't know that before I was part of the study. But frass, you could see that they excrete some of the DON as well as the other mycotoxins that were found in the frass. So showing that they actually take it up, they metabolize it to a certain extent and excrete it. But essentially, the mechanisms underlying the metabolism excretion in mealworms is still relatively unclear, but might be due to microbial breakdown, other enzymatic activities within the mealworm itself. But essentially, what we're able to say is that these mealworms really have appreciably low levels of DON in their body at the end of these 4 weeks on very high contaminated grain. And survival rate was also unchanged between the different treatments. So survival, as you can see, from control to high was unchanged across the different DON-contaminated diets. So that was very important if this is going to be scaled up to something where the insect production is done at a level that is sustainable and being able to use within industry. And many of the other parameters are very comparable between the control and the high DON diet fed mealworms. So no appreciable negative effects on production with high DON grain. And so basically, minimal effects of feeding no value salvage grade wheat, DON contaminated wheat to these mealworms. So this may be a mechanism of adding value to low-graded wheat. It will also decrease the environmental impact of disposing of this low-grade salvage, no-value wheat. And there's actually work being done right now looking at whether or not we can then formulate feed with these mealworms to feed to poultry, generating a novel protein ingredient and providing a feedstuff for production animals. So this may be one of the viable alternatives for mycotoxin contaminated grain. And so with that, I've given you hopefully a little bit of flavor of some of the work, mycotoxin work that's being done. I'd like to acknowledge specifically the two students, now Dr. Anhel Wang, as well as master's student who also has defended his work, Carlos Ochoa, who did the mealworm work, as well as Dr. Fiona Buchanan and Kyla Mader, who are collaborators on the mealworm project, and Karen Schwing-Lardner, who is our animal welfare poultry expert, who helped with a lot of the behavioral work that we did with the poultry studies, as well as industry and funding partners, and then research center staff at the University of Saskatchewan. So with that, I would be happy to take any questions.